So to go back very quickly on your CV, you did your PhD with Michael Arbib at the University of Southern California. You did some modeling work, modeling reaching and grasping execution and observation. So you did a first postdoc with Richard Anderson at the California Institute of Technology, and you did some work on non-human primates, doing modeling, and I think, I don't know if you did it yourself, but on pharmacological manipulation also in non-human primate, yeah. So you did it yourself?
Yeah, well, with a group of people, but yeah.
Okay. So you put your hands in it?
Oh yeah.
Okay. And after that, so focusing on decision-making processing, in this case. And after that, you did a second postdoc at UCL, working with Sven Bestman, who we could see also in January 2019, and Garrett Barnes. And you used in this context EEG and TDCS, and also worked on this decision-making processes. And at the same place, you've been doing some pioneer work, because you've been the very first one who showed that using high precision MEG, it's possible to distinguish between different layers in the cortex, motor cortex. And so you put into evidence some laminar and frequency specific correlates of action selection. So now you've been really successful, because you got a position in Lyon at the Institut des Sciences Cognitives. So you got this position a few months ago.
Yeah.
And right before this, you got an ERC Consolidator grant. And in this grant, the main topic of it is so the sensory motor beta bursts. And you are looking into the mechanisms of the generation of these bursts and their functional significance. And in this project, which is a huge one, as the ERC project are usually, so you plan to do electrophysiology, so invasive recording in non-human primates, and in humans doing non-invasive imaging using MEG and using your headcast procedure. And so I'm going to stop here, and I'm going to let you present your work. Thank you very much.
Thank you. Thank you very much. And thank you all again for having me. Again, it's a shame that I couldn't be there in person. And again, I want to emphasize, yeah, I'll try to keep an eye on the hands, but please let me know if anything requires more explanation. Okay, so one of my main interests is in how the brain produces actions. So how it decides what actions to perform, how it prepares to perform the action once this decision has been made, and how it updates its internal representations based on the outcome of the action. And I studied this mainly using EEG and MEG, which are good for investigating neural activity non-invasively at a fine temporal scale. Now, there are a few classic signatures of motor-related activity that you commonly measure with EEG and MEG. So typically what you see is a burst of high-frequency gamma activity around the onset of the movement. You see a decrease in power in the beta frequency band prior to the movement, and this reaches its minimum around the onset of the action. And this is followed by a beta rebound, in beta activity. So one early idea for the functional relevance of beta activity is that it reflected the idling state of the motor cortex, or promotion of the status quo. So prior to a movement, there's this sort of baseline level of beta activity. The motor cortex is idling. And then as that movement is prepared, this activity is gradually reduced, and it finally reaches a low enough point that the action can be performed. And then there's a beta rebound, sort of reinforcing the new status quo. And now when most people look at induced frequency-specific activity, they start with their time series, right? So this can be sensor data from EEG or MEG, or it can be LFPs recorded with electrodes. And then typically they close their eyes and they perform a time frequency decomposition on each trial. Then they average over all the trials and they open their eyes again. And when you do that, you see something that looks like this. You see sort of average power in several frequency bins over time. And now I should say this is in somatosensory cortex. This is some of Stephanie Jones's work, measured using MEG. And most people then assume that this represents, in each trial, these sustained beta oscillations. However, Stephanie Jones and her group have done some brilliant work showing that if you look in beta power in each trial separately, these don't look like sustained oscillations at all. Rather, it looks like beta power occurs in discrete bursts. And if you identify the timings of these bursts and you average over them in the time domain, you get something that looks like this. It looks like these bursts are caused by these discrete events with a stereotype shape. So up here is showing each individual line is the time course of each burst event. And this is what it looks like when you average over all the bursts. So that was in somatosensory cortex. But what about motor-related beta activity in sensory motor cortex? So inspired by this work, Simon Little and I looked at some MEG data that we collected at UCL using a cued button press task designed to allow subjects to predict the button that they would be instructed to press and to vary the confidence and accuracy of their prediction. So first they see, after fixating, they see a random dot kinematogram. So this is a field of dots moving in random directions with a certain proportion of the dots moving either to the left or to the right. Then following a short delay, they see an arrow pointing to the left or right. And as quickly as possible, they have to hit the left button if the left arrow shows and the right button if the right arrow shows. And importantly, the direction of motion coherence in these dots predicts what direction this instruction cue will be. So if they see most of the dots moving to the right, during the delay, they can be more or less confident that they'll be instructed to make a rightward response. Okay, so we analyzed our data in the same trial by trial way. And indeed, we found that beta power occurred in these transient bursts. And these bursts are caused by discrete events in the time domain with the same waveform shape that Stephanie Jones found in somatosensory cortex. So again, here's showing, this is some example trials. This is showing the time course of each burst, and this is showing averaged over all the bursts. And I have to say, I'm presenting this like it's this nice story, but actually, when I first got this data, before I'd ever seen Stephanie Jones's talk, I did a time frequency analysis, and I peeked at the time frequency representation in each trial, and I saw this, and I thought, oh, I've done something terribly wrong. That didn't work at all. Then I averaged over all the trials, and I found this classic decrease in beta followed by the beta rebound. And I thought, okay, well, that makes sense. And then only later did I see Stephanie Jones's talk that explained what was going on here with these beta bursts, and that might actually be what we should look at. So yeah, when you average over these time frequency data over trials, you see this beta decrease prior to the movement and the beta rebound. So this begs the question, how do we look at beta activity? So if you look at beta amplitude over the course of individual trials, so every line here is showing beta amplitude in this task over a single trial, with time 0 being the response time. This signal is really noisy. It's only when you average beta amplitude over trials do you see this classic beta decrease and rebound surrounding a movement. And if you look at the distribution of beta amplitude over all time points in all trials, so here you see this is the distribution, from each time point in every trial, what's the beta amplitude? You see that the distribution has this long positive tail. And so if you set a threshold somewhere in this tail, like this red dotted line is just showing a threshold and saying, okay, whenever beta amplitude exceeds this threshold, we're going to say that that's a burst. You end up then with a more probabilistic view. So here you can see each point in this raster plot is the occurrence of a high power beta burst. And every line here is a different trial. So you can see that during rest, there's a baseline probability of bursts occurring. And this probability decreases just prior to and during the movement. And then following the movement, there's a very high probability of a burst occurring. So, and in fact, it turns out that this burst probability closely tracks both the pre-movement beta decrease as well as the post-movement beta rebound. So here, this is showing over all the trials from all of our subjects, and each horizontal red line is just showing the boundary of each subject. These green lines are showing the onset and the offset of the random dot kinematogram. So this is aligned to the instruction queue. And each dot again here is a beta burst. And so if you look over all these trials and you compute the burst probability at each time point, you get something that looks like this. And that probability closely tracks the changes in beta amplitude that decrease just before the movement. And the same thing for post-movement beta rebound. So here we've aligned the data to the response time, it's time 0. And you can see that the burst probability is quite low, around 0%. And then it jumps up right after the movement. And this tracks the rebound in beta amplitude that you see. So if we take the view that beta activity is best thought of as bursts that occur with varying probability depending on the motor context, we can identify when these bursts occur in our data and realign the data to be centered on the peak of each burst. So this is showing all the pre-movement bursts identified in a single subject, along with a field map. So this is MEG sensor data. And so when you do that, you can see this dipole reversal from the beginning to the end of the burst. So you have a dipole pointed in this direction, and then it flips back again. And here's the average pre-movement bursts for each subject in our data set, along with the average burst time course over subjects, showing that the shape of beta bursts is highly conserved across subjects. So here, each line is a different subject, and here's the average over subjects. And interestingly, we found the same spatial topography and waveform shape for post-movement bursts, but they tended to have a greater amplitude. So you can see the amplitude of these bursts tend to be a bit greater than these pre-movement bursts. So that was the spatial topography of beta bursts at the sensor level, but what about in source space? And if you look at the spatial topography of the beta rebound in source space over the whole brain, you can see that it's very diffuse. And in many subjects, it actually occurs in the opposite hemisphere. So here subjects were using their right hand to make a response. And in this subject, you see a bit of a beta rebound in both hemispheres. However, if you look at the pre-movement and post-movement beta bursts, their spatial topography is extremely focal. So you see it really only in the contralateral hemisphere. And we can quantify this by looking at how the beta amplitude drops off with the distance. So this is as you move out from the center of the change in beta, what the beta amplitude looks like as that distance increases. And this is normalized to the post-movement bursts. And what you can see is that there's a much steeper drop off here till you get to a full width half maximum for pre and post movement bursts compared to the beta rebound, which has a much less steep slope. Okay, and this just to further illustrate the point, this movie here is showing the time course of the beta activity when you average over trials in a single subject. And so you can see you see this decrease in beta activity. It's really widespread. And then following the movement, you get a rebound in beta activity, but it's really spread out over that hemisphere and even into the other hemisphere. And in contrast, that's a beta burst. Really short, really focal. So we wanted to make sure first that beta was actually bursty rather than an oscillation, because as has been pointed out recently, there are several different possible models that could explain oscillatory activity. So the first is just a pure rhythmic oscillation. Okay, so you have, just this classic sinusoidal oscillation. And this line here is showing basically our threshold for detection. So using whatever experimental technique we're using to record the activity, we can see all of it. And the second is a rhythmic oscillation with time varying amplitude, like we see in the trial average beta signal. And again, here the threshold for detecting can be quite high or quite low, depending on our experimentation. And that might cause what's actually this time varying amplitude to look like bursts, right? Because we can't detect the signal here. Okay, the third is bursts with no underlying rhythmicity, like we see in the single trial beta data. And again, our threshold here could be quite high, or could be lower, but basically there's nothing happening in between the bursts. And there could be a hybrid model where there's an underlying rhythmicity, but we can only see the activity, above this threshold. So here, there's no relationship between the phase of this signal and this signal, but here there's this underlying rhythmic process generating this, but it just looks like bursts to us. Okay, so we're interested in can we distinguish between these last three models? So we did this using a measure called lag coherence. And this is a recently developed measure of rhythmicity that would allow us to distinguish between these models. And lag coherence basically attempts to answer the question, can we predict the future phase of a signal from the current phase? And so the idea is if we have a purely rhythmic signal and there's a pure sine wave of varying amplitude, we can look at the phase at a given time point and pi in this example and say, can we predict the phase several cycles in the future? So if the signal is purely rhythmic, the future phase will be perfectly predictable. And we can see how many cycles in the future can we predict it. Okay? So this would distinguish between these last three cases if we could not predict the phase, because even in the example, the possibility where there's an underlying rhythmicity, but we can only measure above some fixed threshold, that underlying rhythmicity would predict the phase at the future time point. So if we go enough cycles ahead in the future, we'd be able to predict the phase again. Okay, so here this is looking at can we predict the phase 3 cycles in the future? And we did this over frequencies from 5 to 30 hertz. And this is just showing the pre and post movement spectral densities. Okay, so you see this sort of bump around alpha and bump around beta. And this is showing the lagged coherence at each frequency. And you can see there's a peak in lag coherence that corresponds to the alpha peak, but really it drops down around beta, and there's no peak there in lag coherence. We also said what would happen, just to get a sort of idea of the null distribution, what would happen if we took the data and shuffled the cycles. And so this is just showing that when you do this, it doesn't really affect the power distribution in different frequency bands, but you get a flat lag coherence. So you destroy that ability to predict the future cycle of alpha. But for beta, it was already pretty flat to begin with. Okay, and so that was just 3 cycles in the future. But remember we said we could see, how many cycles in the future can we predict the phase? Okay, and this is just showing that for pre and post movement beta activity. And for both of them, you can see the sort of peak around alpha that slowly deteriorates. So around 4.5 cycles, 5 cycles, you can no longer predict it. But post movement, it goes quite far. Whereas for beta, it's quite flat. And we see that in the cycle shuffle data as well. Okay, so how are these bursts generated? So one prominent computational model simulates beta bursts using a biophysical model of a cortical column that involves a network of pyramidal cells in the deep and superficial layers, as well as inhibitory interneurons, which are not shown here. And this model is driven by 2 temporally aligned inputs to each cortical layer. So you've got this proximal drive that targets the deep layers, and it consists of this weak, temporally dispersed pattern of activity. And then the distal drive targets the superficial layers and is stronger but briefer. Okay. Now, the proximal drive causes current to run from the deep layers in the direction of the superficial layers, and the distal drive results in this stronger current running in the opposite direction. And the combination of these two currents gives you a net cumulative dipole with the same wavelet-like time series that we found in our human subject data. And what's interesting, and it would be very relevant for the paper that the students have read before this, one of them, is that if you do a time frequency decomposition on this signal, on this time series, you end up with a burst of beta activity, just like we saw, burst of beta power, just like we saw in the individual trials in human subjects. So again, this is showing that if you apply a Fourier analysis to any signal, you'll get power somewhere. And the fact that you get power in the beta frequency range here is sort of an accident of the shape of this time series. So it doesn't mean that there's an ongoing oscillation. And there could also be other time series that would give you a similar shape in frequency space. And this model is nice because it makes some really specific predictions. So namely, if we could measure the time course of laminar activity in a patch of cortex where a burst is occurring, activity should be strongest in the deep cortical layers at the beginning and the end of the burst. So that's when you're only getting this proximal drive. And it should be strongest in superficial layers at the peak of the burst. So that's when this strong distal drive is sort of overwhelming or disrupting this weaker proximal drive. And it would be nice if we could test this prediction non-invasively in the human brain. So magnetoencephalography, or MEG, is an attractive technique to test these predictions because it's a direct measure of neural activity. It's non-invasive, it covers the whole brain, and it has excellent temporal resolution. But when we do that, we're just measuring activity in these sensors. And what we really want to do is reconstruct the source, the cortical source of activity measured from these sensors. And in this type of research, this is known as the inverse problem. Okay, so getting your sensor data and projecting that back into the brain. And this is a difficult problem because it's known as an ill-posed problem. And one of the reasons is that there's many more possible sources of activity than there are sensors, right? The brain's a big place. It's got a lot of neurons. There's many different possible locations that you could have activity in. And there's only, a few hundred sensors. Okay, so to solve this problem, what we do is we build a generative or forward model of how source activity translates to measured activity at the sensors. And this is because, these sort of networks of pyramidal cells, they have electrical current, and these currents generate a small, tiny magnetic field. And so the forward problem, or going from how this electrical activity translates to magnetic signals that we can measure at these sensors, is very easy to do. Okay, that's a tractable problem. So we build some assumptions into these forward models. And one assumption is that synchronous activity in nearby pyramidal cells sums up to give us a measurable signal. We use individual subject anatomy. So we take an MRI, a high resolution MRI of each subject, and we use that to construct a forward model predicting how source activity at each cortical location in that surface mesh gives rise to sensor signals. And then we can use this generative forward model to make predictions. So we can say, for source activity in different locations, what would that sensor activity look like? Okay, so here's our measured data. We can say, okay, what if the activity was here? Okay, using our forward model, that's what the sensor data would look like. Okay, what if the source activity was here? We put it to the forward model, that's what the data would look like. And then we can compare these different forward models, these different predictions to the measured data to determine the most likely source of source activity. So we need a generative model of sensor activity that includes representations of deep and superficial cortical lamina if we want to test this model, right? If we want to test its predictions. So to build this type of forward model, what we do is first do an MRI on each subject, and we extract the peel and the white matter surfaces. Okay, so the deep and the more superficial surfaces. And then we constrain this electrophysiologically. So we use the fact that cells in layer 2 and 3, these superficial layer cells, will contribute the most to the cumulative dipole moment on the peel surface. And these deep cells, these layer 5 cells, pyramidal cells, will contribute most to the cumulative dipole moment on the white matter surface. And then we constrain the orientation of these cells to be oriented in the same way as cortical columns. And finally, we include this notion of recurrent connectivity amongst these cells. So we model activation not just as a single dipole, but a patch of activity with some spatial extent. Okay, now we can use these generative models to determine the most likely laminar source of measured activity. And this analysis fits one generative model that assumes that all the activity was happening on the peel surface. And then we fit another model assuming that it all occurred on the white matter surface. And remember, these are representing sort of two hypotheses, that the activity is happening superficially or happening in deep layers. And then we can compare how well these models fit the two data sets, the same data set, sorry. The two models fit the same data set. So the fit metric that we use is model evidence, and specifically the relative evidence of the two models. So in the plots that I show you, we're plotting the difference in model evidence. And so that's the model evidence for the peel surface minus the model evidence for the white matter surface. And so whenever this value, this difference is positive, it means that there's more evidence for the activity occurring in the superficial layers. And wherever it's negative, it means there is more evidence for the activity having occurred in the deep layers. Okay, so first we use this model comparison analysis to determine the laminar origin of those induced motor signals that I mentioned at the beginning of the talk. So the burst of gamma at the onset of the movement, the slow decrease in average beta power prior to the movement, and the beta rebound. And so our first step is to fit each generative model to the data. So here you see the decrease in beta power on the peel surface and the white matter surface. So from these two models for a single participant. And here's the distribution of our peel white matter comparison over all the vertices on each surface. And you can see that this is skewed negatively, so skewed towards the white matter or the deep surface. And we found that this pattern holds up over all the subjects. So here showing this comparison over the whole brain for each subject. Each bar here is a different subject. We found the same thing for beta rebound. So if you look, you see this increase in beta power on the peel and white matter surface. But over all the vertices in one subject, this is skewed towards the deep layers. And we found that for all of our subjects. But interestingly, for gamma, we found localization to the superficial layers. So we had a sort of similar topography here of gamma increase, but here this distribution is skewed positively. And for every subject but two, we found that gamma localized to the superficial layers. Okay, so we know that on average, beta activity localizes to deep cortical layers. But what we really want to do is look at the time course of laminar activity during beta bursts. And we want to do this at a fine temporal scale. So what's nice about these discrete burst events is that we can average over them and treat them as an evoked response. So we averaged over these events at the sensor level. We used our laminar generative model, where we have one model representing the superficial layers and one representing the deep. And we ran a sort of localizer, source reconstruction using only the peel surface on the entire averaged evoked response. Okay, so we found the peak location in cortex where this burst is occurring. And then we used that location on the peel surface and the corresponding vertex on the white matter surface as priors in a sliding time window source reconstruction. Okay, so to do that source reconstruction, we ran this model comparison within a small time window of the average evoked response. So in this time window, is there more evidence for the Peel model or the white matter model using these priors? And then we can slide that time window along and plot the difference in model evidence between the Peel and white matter generative models. Okay, so before we run this analysis on our human subject data, we wanted to see if it would even work in principle using this model. So we generated some simulated beta bursts using this biophysical cortical column model. And we ran these simulated source signals through a generative forward model to create a synthetic data set. Okay, so here's a MEG sensor data just generated from this model. And then we ran our sliding time window analysis on the data set. And when we do that, we get that activity is strongest in the deep layers at the beginning and end of the burst, and strongest in the superficial layers at the peak of the burst. Okay, so just as you'd expect. And this is just showing what this beta burst activity looks like at the source level. Okay. So just as we predicted from the model, at the beginning and the end of the burst, you're only seeing this proximal drive in the deep layers, and that's where activity localizes. But then at the peak of the burst, you get this strong distal drive to the superficial layers, and that causes the activity to localize superficially. And we also ran, we really wanted to be sure about this, so we ran a bunch of control analyses where we switched the proximal and distal drives, some that included only one drive, one where we switched the orientations of the two dipoles, and the results were as you'd expect. If you flip these two, the location of these two inputs, this flips upside down. If you only include the proximal drive, the whole thing localizes to deep layers. Okay, so we're pretty confident that the analysis would work, at least with the simulated data. And here's just showing some of the control analyses that we did. Yeah, so here's showing what happens if you flip the location of these two inputs. You get that activity localizes superficially at the beginning and end of the burst and to deep layers in the center. If you include only the distal drive to the superficial layers, you get activity localizing superficially. And with only the proximal drive to the deep layers, you get activity localizing to deep. Okay, so now we're ready to run the analysis on the human subjects. So this is showing the peak of the localizer source reconstruction for each subject for pre-movement beta bursts. And you can see that these are tightly clustered around contralateral sensory motor cortex. Here's the pre-movement beta burst source time series for each subject. So that's each colored line with the average shown in black. And here's the results of the sliding time window analysis on human subject pre-movement beta bursts. So just like predicted by the model, you get activity localizing to deep layers at the beginning and end of the burst, and to superficial layers at the peak of the burst. And again, we wanted to be sure about this, so we ran some control analyses where we shuffled the data. So in blue here is where we shuffled it temporally. We shuffled the time series of each beta burst before averaging them. And we also ran a spatial control where instead of using the priors from our localizer reconstruction, we ran it at random locations in the brain. So where we didn't think beta bursts were occurring. And when you do either of that, you get this sort of flat prediction, not biased towards either layer. And the results for the post-movement beta bursts were largely similar. And these are higher power, but you get the same pattern of localization to the deep layers at the beginning and end, and the superficial layers during the peak. Okay, so now we know something about how these bursts are generated. We have this mechanistic biophysical model of burst generation at the level of cortical microcircuits, and we tested the model non-invasively in human subjects and found good support for it. But what are these bursts doing? So if we go back to our probabilistic view of beta bursts, here's the timing of each burst again in each trial across all of our subjects aligned to the movement offset. If you look at the probability of a burst occurring at any given time, you can see that the probability decreases prior to the movement and jumps back up after the movement. But if you try to quantify when and how many bursts occur in a given trial, you get less than 1 on average prior to the movement and about 1 after the movement. And that burst after the movement occurs about 3/4 of a second afterwards. So what's going on here? This is really difficult to reconcile with the idea that the beta represents an idling motor cortex or promotes the status quo. How could it be if you have, about 1 burst prior to a movement and 1 burst after the movement, and it can happen up to half a second later or more? So we get a clue about the functional role of beta bursts if we look at the burst timing and probability relative to the subject's behavior. So in this task, we have 2 behavioral dimensions. We have the response time and the accuracy. And if we look at pre-movement beta bursts, here looking at the difference in timing between the final burst prior to the instruction cue, we see that the timing of the final burst is later in probability in slow trials prior to the cue. However, there's no relationship with accuracy. If we look at post-movement beta bursts, we find no relationship with response time, but the first post-movement burst occurs earlier and there's a higher burst probability in correct compared to incorrect trials. Okay, so now we have a clue. Pre and post-movement beta are functionally dissociated and they're closely tied to response time and accuracy. And importantly, we found that burst timing was a better predictor of behavior than beta amplitude or burst rate. Okay, so this is when you make one model that is trying to predict the response time, or in this case, predict the accuracy using the beta amplitude, the mean beta amplitude within a time window, the burst timing, so the timing of the first burst after the timing of the last burst prior to the instruction cue, or after the response in this case, or the burst rate, so the number of bursts that occur in a time window. And we can compare these models and show that whether you use different metrics, AIC or BIC, the best model is the one that includes burst timing, followed by burst rate. And this is all relative to the beta amplitude, which was the worst. So one hypothesis that would be compatible with these results is that beta bursts reflect an activation of motor goal representations. So they need to be activated in order to prepare an action, and therefore the later this occurs, the longer the response time. And they also need to be activated following an action to update internal motor models, which may be delayed after an error that requires more sensory motor integration and information gathering. Okay, so in summary, I tried to hopefully convince you that sensory motor beta is composed of discrete transient bursts, that distinct deep and superficial layer inputs drive beta bursts. And as a side note, it's now possible to test different laminar theories non-invasively. So one thing I'm interested in doing is applying this time-resolved laminar analysis to evoked responses as well. We found that burst timing was more predictive of behavior than average beta amplitude or burst rate. And I want to argue that this suggests an active information encoding role for beta rather than simply inhibition. And so, yeah, I apologize that really this talk was a bit more finalized version of some of the results that I showed you last time. And so now I just want to give you an idea of some of the work that I'll be doing now in the future in the new team that I'm setting up at the ISC. This is the Decision, Action, and Neural Computation team. And there are several projects aimed at testing this hypothesis from different angles. So in an ERC-funded project, I plan to determine the computational mechanisms driving beta bursts. So this would involve developing a multi-scale computational model, embedding this cortical circuit model within a larger network, and doing some functional connectivity analysis on beta bursts to determine which other brain regions project to motor cortex to drive beta bursts. And this will be used to constrain the connectivity of this model. And then I want to hook that up to a generative movement model to make some predictions about how beta bursts could influence movement kinematics. Second project is to determine the functional roles of beta bursts. And this is going to involve a MEG study with human adults involving a visual motor adaptation task to try to look at the role of beta bursts and beta activity in implicit and explicit adaptation. And also analyzing some neurophysiology data from monkeys performing reach and grasp movements to look at beta bursts in motor cortex in relation to population activity in premotor cortex in some low dimensional subspace. And then finally, what I think is one really interesting part of this project is a longitudinal study with human infants to look at how beta activity affects motor skill maturation. And so one striking feature of this model is that the two inputs to the deep and superficial layers have to be aligned temporally. And if you run the model with one input arriving too early or too late, you get a burst of activity at a lower peak frequency. And because the timing of these inputs could depend on myelination of axons and tracks, fiber tracks in the brain, I want to use DTI to track in human infants in their first year of life how these fiber tracks develop. And at the same time, or at the same ages, perform an assessment involving EEG and behavioral reaching and grasping and tracking the infant's kinematics. Basically to look at how these changes in anatomical activity drive maturation of infant beta activity and how that relates to their developing motor skill. And then finally, involved in an ANR funded collaborative project with some colleagues from the CRNL and Grenoble, with the goal of applying this sort of trial by trial analysis of frequency specific signals to developing non-invasive brain machine interfaces for rehabilitation. Okay, with that, thank you all very much for your attention. I just want to thank collaborators and funders, and yeah.